So thank you very much. Thank you, Rob, and to the entire ERGA for this invitation. Indeed, we are sisters and brothers as nodes of the Earth Biogenome Project. And I would just want to tell you about our experiences in setting up this African Biogenome Project, and perhaps we can see how we can share experiences and help each other move along. So I'm Anne, I'm chairing, and I have my co-chairs, Dr. Thangod and Ntanga from South Africa, and Professor Jikeng, who's the DG of the International Livestock Research Institute. So I will very quickly give you highlights of the project and give you some plans that we have for the future, and also show you some of the sequences that, some of the genomes that we have sequenced on the continent. So let me start by just giving you some of the goals of the African Biogenome Project. These are the three main goals: that we want to coordinate all non-human genome sequencing initiatives so that anyone who's sequencing eukaryotes, that we're able to get in place and offer technical advice so that we can be able to do it to the standards that it will contribute to the African Biogenome Project. And we also want to have a forum for discussion about what sequencing means for all the other genomes that are non-human. And why non-human? Because there's another initiative that's sequencing African human genomes. So since the human genome is taken care of, we want to take care of all the others. We also want to initiate some non-human genome sequencing efforts. And our goal is to sequence over 100,000 genomes in the next 10 years. More importantly, we want to use this data to contribute to regional biobanks and also start African repositories for data and samples on the continent. We also want to very, very much inform policy, and extremely important for us to build capacity as we go along. I mentioned that we are sequencing all non-human species, but let me just clarify that it's all eukaryotic lineages, because there are groups such as the African CDC that are sequencing prokaryotes to do with disease and vectors and things like that. So we want to move away from that and sequence eukaryotic lineages. I mean, the task is huge. And to do this, what we did is that we had a questionnaire that was distributed around 300 institutions in Africa, encouraging scientists to nominate their species and to give the reasons why. Through these nominations, we were able to find recurrent themes, and those themes now formed a basis for us to shortlist the first species that will be sequenced. For instance, the species is contributing to food security. Maybe it's an African indigenous livestock or plant that is eaten. Maybe it's completely endemic to Africa. Maybe it's only found on the African continent. Or maybe the habitat is being ravaged and destroyed rapidly, or it's facing threats of extinction. So using this criteria, we're able to narrow down into some of the species, the first 100 species that we'll be sequencing. What do we envisage? We envisage to build more Africa to Africa collaborations. The Africa to North Europe and the rest of Europe, Americas, Asia's, those collaborations have been very, very strong, and Africa as a whole has indeed benefited. But what has not happened effectively are conversations within Africa. So we don't know who is doing what around the continent, and we want to increase this conversation so we can be able to even get to know each other, because we didn't know each other. And through this, we'll contribute to the genomic building of genomic resources in Africa. And as I have mentioned, inform and drive policy and build our socioeconomics on the continent. Africa Biogenome, the conversation about it started in 2019. You'll be surprised that the first virtual biogenomics conference was the time that we met. We met online virtually as Africans on that conference, Biodiversity Genomics, of October 2019. And that's when we started speaking to each other. And those conversations continued again. We had a poster session during the next conference, and we got to know more Africans who are dealing with biodiversity and genomics. And that helped us build a momentum towards the launch of the African Biogenome Project in September of 2021. All from that time to last month, we were undertaking the pilot project, and this was the one that was establishing our framework, our policy documents, our sequencing pipelines, and we were able to close that with a total of 13 genomes. Our project aimed to sequence 2,500. We're not yet there, but at least we have begun. And going forward now, we want to move into large-scale sequencing of both plants and animals as we move along. We have 4 programs within the African BioGenome Project that will address this challenge of sequencing. The first is the Nelson Mandela Africa BP Genomes for Conservation of Nature. And through this, the core objective is to sequence 60,000 animal species that are indigenous to Africa. And we have developed partnerships with the University of South Africa, the Rockefeller University, through VGP, who have many of their species also being found on the African continent. We have identified a technology partner. ERGA Biotech is a South African company that has installed a PacBio, and University of Cape Town is partnering with us with the South African repository, the South African Biodiversity Institute, that has been able to donate samples for sequencing. We also have the DAISY Africa BioGenome Grassroots for Conservation. The core objective here is to sequence 45,000 plant species across the African continent. UNISA is a key player in this, together with Beijing Genomic Institute, South African Medical Research Council, the University of Cape Town, and the Southern African National Biodiversity Institute. So we identified plants that are on the 10KP project, and these plants are indigenous to Africa, some of them, and we were able to develop a collaborative framework with 10KP, and we are now going to sequence some of their plant species on the African continent at the South African Medical Research Council. It was interesting to hear the update about on the ELSI committee, because that's something we also realized, that on the African continent, we needed to have conversations about DSI. We needed to talk about the Nagoya Protocol, the post-2020 biodiversity framework. And to be able to bring us together, we started roundtable meetings that occur once a month. They happen throughout 2022, 2023, to be able to discuss what access and benefit sharing means for different regions, because the laws are crafted differently and their implementation are different across the board. And then in October 2020, we launched the Africa BP Open Institute for Genomics and Bioinformatics. This is our training and capacity building arm, and I shall be discussing more about this later. So let me give you some of our highlights. We published our position paper in 2020, and this is where we put out our goal of sequencing 100,000 genomes. It was received very, very well throughout the world, and thank you to the entire scientific committee for welcoming this African initiative to sequence plants and species on the continent. Again, the profile was lifted because I think there was a bit of surprise that this project was being started in Africa, but after the surprise wore off, a lot of rallying of support from agencies like UNESCO, FAO, and all this, to be able to see how we can be able to get this project off the ground. So our aim is to sequence 100,000 genomes in 10 years, and we aim to sequence most, in fact, we're saying between 80 and 90% of the genomes on the continent. That will obviously generate a lot of data. Yeah, we've estimated at least a minimum of 200,000 gigabases of DNA sequences. And we wanted not only to sequence this on the ground, but also to be able to have the African early career scientists do the sequencing and annotation on the continent. And immediately we embarked on this, we realized that we had a big gap. And the gap was in terms of institutions that had the capacity to do the sequencing. And we also had gaps in terms of scientists, African scientists who were able to do the genomics and bioinformatics sequencing and assembly on the continent. And therefore, this is the reason behind the African BP Open Institute, to be able to bridge some of these gaps. So we launched this in 2022 with a position paper that was published in Nature Biotech in September of last year, where we outlined our goals of how we're going to bridge the gap in terms of African biodiversity and genomics. The first way was holding regional workshops. Regional workshop meaning that we wanted to bring genomics and bioinformatics closer to the early career scientists by going to where they are located. So we had 4 workshops, one in Nigeria, and that's the image that you see here. That was in February of 2024. And following the success of that, we modeled that success to be able to launch workshops in South Africa that happened in August, in Northern Africa that happened actually in Morocco, and Northern Africa that happened in October of 2023. And East and Central Africa, the workshop happened in December of 2023. These workshops were attended by 3,000 early career scientists. Each workshop had 200 slots for in-person attendance, and they were always oversubscribed by 10 or 15 in person, and then the rest of them attended virtually. And then following this, we had 350 slots for practicals, and these were we received over 4,000 applications for these 350 slots. So you can see the demand for capacity building and training. So the conference followed a style of 5 days. The conference was held over 5 days with 2 days in person and virtual attendance. And there were plenary sessions from different speakers trying to talk about different matters, all the way from sampling to sequencing and annotation. And these 2-day conference was followed by practicals. These practicals were offered for free. The only contribution for the young early career scientists was for them to meet their cost of accommodation and transport, and we met the cost of the teas and the lunch during the day. So this happened over 3 days, and we count now that we trained over 381 early career African scientists on hands-on genomics and bioinformatics. So in the application form, they would fill what they are interested in, whether they want to go to a wet lab, a dry lab, whether they were interested in sampling. And we configured practicals around this. And each practical session held small groups of not more than 15 to 20 participants. And African BioGenome Project actually leveraged heavily on contributions from technology companies. So technology companies like Impakaba Biotech and the ones written here, Separations in South Africa, provided the reagents, the infrastructure, the laboratory, they paid the bench fees. And therefore our contribution was only just to provide the resource people, and the young early career scientist provided with his own transport and or her own accommodation. And I just want to show you, this is the practical that was held, 1 of 5 that was held in East Africa, 1 of 7 that was held in South Africa, 1 of 5 again that were held in Nigeria, in Algeria, and in Northern Africa. So they were very, very informative, and we trained at the level at which the African scientists needed. You can see here, this is just a pipetting class, because I mean, genomics, sometimes they've only seen pipetting online, and maybe they've never actually been able to aspirate something. So we started from the basics, and we hope to build on that basics as we go along. And again, Central Africa has been really difficult for us to reach as African BioGenome. And we were happy to be able to have 3 practicals running in Central Africa. And that means that we'll be able to now quickly begin a node of young early career scientists in Central Africa. Here we can see some from University of Yaoundé, and DRC Congo, as you know, is right in the middle of Africa. And the workshops were very well received throughout the continent. And you can see the Dean of the Faculty of Sciences in Morocco saying that it's not only an honor for Mohammed V University to host the conference, but also a solemn responsibility for Africa. And this is what we have seen, that now we have African institutions opening their doors, giving us facilities for conferencing, for labs at minimum or no rates for us to be able to hold these workshops. Participants leave with a certificate for participating, and if they also attended the practical session, they also get a certificate for the practical session. And these certificates have been very, very useful and a strong sense of motivation for the young early career scientists. What is unique about the Africa BP workshops, that they're open, accessible, no fees are charged. They have blended participation, allowing for an increased number of Africans to be able to participate. They're hosted by African institutions. So again, we're keeping it on the continent. And because of the participations of scientists from across the region, we are able to meet and have a discussion of what it means to train in terms of genomics and bioinformatics. Again, they're being held in all the 4 regions of the African continent, so the training is being taken closer to the Africans. And the workshop presents an opportunity for African institutions to build capacity, and not only that, to be able to see the numbers of young early career scientists who are present, and therefore begin to consider making investments in terms of purchasing equipment and building relevant infrastructure. So our plan is to publish our findings for the 2023 in a manuscript. This manuscript has been submitted, but we already have a preprint online to be able to share with the world what our lessons are in how we can be able to establish genomics and bioinformatics at the grassroots level. We have sequenced some genomes, a total of 13. Pictures are shown here, and I'll go in further and show you our model. So this group of 13 species have been sequenced with the support of University of South Africa. They provided the funding, they provided the infrastructure, the human resource to do the sequencing, and they paid the technology company Inkaba that has the PacBio that did the sequencing for us. As you can see, the samples are drawn from different material, and we have very, very good coverage for the particular species. These 3 species were on the VGP species list, and we were able to find them in the South African biobank and be able to carry out the sequence. And again, you can see this coverage is quite high. We're talking about some of the challenges we are also facing in terms of sequencing of plants. This is one, we are going to resequence it because, we need to get additional data. But this is on the 10K list and was sequenced at the South African Medical Research Council. So what are our plans? Again, we plan to continue with a workshop series across the 4 areas of the continent. The students who attended the first phase will move into a new phase, an advanced phase, and we hope to get fresh early career scientists who we can begin again with at the grassroots level. We have also established an Africa BP genomic Fellow for Genomic Leaders. This is in partnership with Carl Woese Institute for Genomic Biology in the US. And it's a fellowship that we have just recently announced, actually about 2 or 3 days ago. Our 10 early career scientists who are able to meet our criteria for the shortlist, and they come from 10 different regions. So these students will spend some time in Morocco. They will begin their biogenomic and bioinformatics training on the ground. We have already identified the next crop of Africa biogenome species that will be sequenced, and we think that the best way to go is to use that training ground as a way to also double up as our sequencing effort towards this particular initiative. And as I close, let me just give you some food for thought as we have run this sequencing effort and African Open Institute for about 2 years now. What have we learned as we have gone along? That we can sequence high quality genomes on the continent. And it comes at a cost, yes, but sequencing off the continents solves a very short-term goal in the sense that if you're comparing dollar by dollar, how much it will cost in Africa and how much it will cost in France, for instance, of course, the French cost will be much cheaper because the only perhaps thing that you're going to pay for is the shipping out of the sample, the freight costs. But in terms of the cost to Africa, it has a much longer and higher cost because it goes against the long-term consequence of building capacity for the African continent. So Dr. Thank God, my co-chair, raised this question: how many scientists easily ship samples out of the continent but are not proud of it? And how many scientists put in their short-term personal interest before the long-term African gain? So if I'm in a hurry to sequence something, it's very easy for me to ship it out of the continent, get it sequenced, get my data and publish. But I'm fulfilling a personal interest against a longer goal for Africa that we should be able to build capacity on the continent. So how many of us, including us African scientists, complain about limited capacity in Africa and yet fail to lay the foundation for the future? Because as we continue to ship out samples out of the continent, we continue to make the instruments that have been purchased at great cost on the continent lie idle. And also we fail to use that capacity that is there. And perhaps very few institutions will be willing to put in that money if that capacity is not being utilized. So also another food for thought is that training of African early career scientists needs to go hand in hand with mechanisms of retaining them on the continent. It's very good that we have institutions out of the African continent, in Europe and Africa, in Americas and elsewhere, that have offered opportunities for African scientists to train there. But what perhaps may be lacking is how this African can be transferred. Because if we are unable to think about them and what will happen to them when they come back home, well, they come back home to low salary paying positions, poor infrastructure, and no opportunities for promotion and growth, and they feel underutilized. And at the next opportunity, they will be very quickly shipping themselves and their families out of Africa. So training of African career scientists needs to go hand in hand with an additional step of seeing how this capacity that has been developed can be transitioned into Africa. If this had happened over the years, perhaps it would not be so difficult to get people who are hands-on in terms of genomics and bioinformatics on the continent. So we recognize the important role of the African Open Institute, and through this, we are hoping that we can now bridge the gap in terms of training and also discuss what capacity building and training needs are needed for institutions around the continent. And also, more importantly, to discuss curricula that needs to be developed that need to go into our local universities to be able to bridge this gap. And I thank you and welcome you to visit our website, and I welcome any questions that you may have. Thank you very much.